Notable guest, ladies and gentlemen, please be seated.
We wish everyone a very good morning. Dr. Moktazul Haizat Mokhtaram, Deputy Dean of Academic, Associate Professor Dr. Nur Faizul Hadri bin Nordin, Keynote Speaker, International Institute for Halal Research and Training, IIUM.
Dr. Roshaini Binti Othman, Head of Department of Science and Biotechnology, T.S. Dr. Noor Azian Binti Shari, Head of Department of Engineering, Dr. Yashoda Sundaraj, Program Director, esteemed board of panels, respected guests and participants. On behalf of the committee members, we are overjoyed to welcome you to the
Faculty of Engineering and Life Science Undergraduate Symposium FELS-US 6.0. Ladies and gentlemen, we will be your hosts for today. I am Yogasri and my partner.
Hello everyone, I'm Charulata. We are absolutely thrilled to be here. This will be our sixth symposium in the series, with the theme Emerging Science and Engineering Towards Future Global Challenges.
Among the aims of the symposium is to provide a platform for sharing of undergraduate students' research discoveries, refining leadership and presentation skills. We hope it serves as a medium of interaction and network for the research community in science, technology, and engineering.
We trust that attendees will find the session welcoming and productive to all. Ladies and gentlemen, before we move further along the program, we would like to invite Dr. Muhammad Rafi bin Soleh to lead the Dua recitation.
Bismillahirrahmanirrahim. Alhamdulillah Rabbil Alamin. Wassalatu wassalamu ala Sayyidina Muhammadin wa ala alihi wa ashabihi ajma'in. Ya Karim, Ya Wadud, sesungguhnya kami berhimpun pada hari ini bagi menyatakan kesyukuran di atas kurniaan-Mu yang tidak ternilai. Jadikanlah kami hamba-hamba-Mu yang sentiasa bersyukur, sama ada nikmat yang sedikit, lebih-lebih lagi nikmat yang melimpah ruah kepada kami. Ya Allah, Ya Rahman, Ya Rahim, Engkau berkatilah dan rahmatilah perjalanan majlis FELS Undergraduate Symposium pada hari ini. Sesungguhnya Engkau maha mengetahui bahawa kami di sini ingin menimba ilmu-Mu yang sangat luas bagi mengembangkan ilmu pengetahuan kami sebagai hamba-Mu yang Engkau redai. Justeru berkati dan rahmatilah perhimpunan kami ini. Kurniakanlah kepada kami ilmu-Mu. Sinarilah hati-hati kami dengan cahaya dan hidayah-Mu. Semoga dengannya kami mudah menerima dan menghayati serta mengamalkannya. Ya Zal Jalali wal Ikram, kurniakanlah jua kepada kami kesihatan anggota, kecergasan minda, ketenangan jiwa, kekuatan semangat. Semoga dengannya kami mampu mendepani cabaran, berdaya saing dan mampu mengangkat martabat diri, keluarga, masyarakat dan negara. Rabbana atina fid dunya hasanah wa fil akhirati hasanah wa qina adzabannar. Wa sallallahu ala sayyidina Muhammadin wa ala alihi wa sahbihi wa sallam. Walhamdulillahi rabbil alamin. Terima kasih. Wassalamualaikum warahmatullahi wabarakatuh.
Thank you, Dr. Rafi. We now invite Dr. Muqtazul Haizad Mokhtaram, Deputy Dean of Academic, Faculty of Engineering and Life Sciences, to deliver the welcoming speech and officiate Faculty of Engineering and Life Sciences Undergraduate Symposium 6.0.
Assalamualaikum warahmatullahi taala wabarakatuh, and good morning. Okay, first of all, I would like to thank everyone for attending this final year project symposiums of the Faculty of Engineering and Life Sciences. Your presence is greatly appreciated and shows support for the efforts and dedications of our students. This symposium is an important platform for our students to present the research and innovations they have worked with full commitment. It is proud achievements not only for the students but also for the entire faculty. The project presented today reflect the high quality of education and research at the faculty, as well as the vibrant spirit of innovations among our students. To the students, I would like to congratulate you all for your efforts and achievements. You have shown diligence, creativity, excellence in carrying out your projects. I'm confident with this experience will serve as a strong foundation for your future success, whether in academia, industry, or any field you pursue. I would also like to take this opportunity to thank all the academics, advisors, the industry advisors who have provided guidance, support to our students. Your role is crucial in ensuring this project runs smoothly and achieve the level of excellence we witnessed today. Thank you also to the lecturers and all members of the faculties who have worked hard to make this symposium a success. Your efforts are greatly appreciated and ensure that this event can be carried out smoothly and successfully. Lastly, I hope that this symposium will inspire all students to continue striving for excellence in whatever fields they pursue. May we all benefit from the presentations and discussions that will take place and continue to strive to advance ourselves and our faculty. With this, I'm pleased to officially open the final year project symposiums of the Faculty of Engineering and Life Sciences. Thank you very much.
Ladies and gentlemen, we give you FELS Symposium montage video.
5, 4, 3, 2, 1.
and officiation. To the viewers on FB Live,
our distinguished keynote speaker, whose work has significantly advanced our understanding of halal science and biosecurity, Associate Professor T.S. Dr. Nur Faisal Hadri Nordin, presently holds the position of Deputy Dean of Academic and Internalization at the International Institute for Halal Research and Training in HART. He is also the chairman of IIUM Institutional Biosafety Biosecurity Committee. Presently, Dr. Faisal serves as the editor of the Halal Sphere Journal and holds editorial position in several esteemed local and international peer-reviewed journals. Dr. Faisal has been recognized as a professional technologist (TS) by the Malaysia Board of Technologies, specifically in the field of biotechnology. His work spans over various domains, including environmental biotechnology, halal science, polar science, and biosafety, biosecurity. He actively engaged in community outreach and knowledge transfer projects, particularly focusing on promoting integrated and sustainable agricultural practice within the framework of halal and Toyib lifestyle principles.
please join us in giving a warm welcome to Associate Professor T.S. Nur Faisal Hadri Nordin as he shares his insights and experience with us.
Thank you, MCs. Okay. Dr. Haizat, okay, the Deputy Dean of Research, yeah, and Dr. Yasota, the Program Managers, Head of Departments, colleague lecturers, and also students. Assalamualaikum warahmatullahi wabarakatuh, and very good morning. Yeah. All right, so thank you for inviting me as a keynote speakers. Okay, I was here in like some introduction. I was here in 2004 for 3 months as a staff of UNISEL. Okay, before I moved to IIUM. Okay, and pursue my PhD there. All right, so for today's lecture, all right, I will. Okay, I will give a lecture on frozen treasures. Okay, revealing the potentials of cold active bacteria. Okay, this is my project with Yayasan Antarctica Sultan Mizan. Yeah, together with Chilean Navy. Right. So this is the topics. Okay, so I intentionally give these topics because I want to attract the students. Yeah, so that we know that we can go more. Okay, not only in the lab, but we can go further. Okay, even to Antarctica, where no people, no inhabitants around there. All right. I control good. All right. So for my projects, yeah, for this project, for this work, I'm focusing on cold active bacteria. We know that we have the mesophilic at normal temperature, okay, psychrophilics at cold temperature, and also thermophilics at cold temperature. Okay, so this project focus on cold active, okay, psychrophilic, okay, and also psychrotolerance. Okay, and but more on this project is psychrotolerant because the intentions of the project is to actually use it for industrial purposes. Yeah. All right. So if you want to see what is the difference between psychrophiles or psychrophilics and psychotropes, okay, psychrophilics, uh, the optimum temperature is 15 degree and below. Yeah, and psychotrope zero. Sorry, optimal temperature is 20 to 40 degree, okay, but it can grow at low temperature, 0, yeah. So the minimum and maximum temperature for psychrophiles or psychrophilics, minimum is 0 or lower, and maximum is below 20 degrees Celsius. Yeah. So that's why I want to use the psychrotolerance, yeah, so that we can actually easy to handle in the industrial purposes. All right. So what is the strategy that all these psychrophiles or psychrotolerance, okay, can live, okay, in the cold temperature, lower temperature? Yeah. Number one is the cellular adaptations. Yeah. I won't go in detail about this, but this is just the informations. Yeah. Regulations of the cell membrane fluidity, okay, for cellular adaptation, carotenoids productions, and also cryoprotectants. Yeah, they produce the cryoprotectants so that all the machineries, cell machineries, is protected, so then they can function. Okay, and in terms of molecular adaptation, where they produce or express a different genes. Yeah, maybe same genes but different. There are some different in terms of at the temperate genes, okay, or the enzyme, something like that, but it's different a bit in terms of the molecular adaptation. Okay, chevrons, okay, proline isomerizations, and also the protein and enzyme is a bit different. Yeah. All right. So, what is the benefits of this cold active enzyme? Yeah. First, it's highly efficient catalytic activity at low temperature. If we use the normal enzymes, okay, so we need to supply the energy, the heat, okay, so that around 30, 32, something like that. So we need to actually give the energy, okay. But this one can work at low temperature, right? So especially if the cold country, yeah, like four season countries and all that, okay, it's actually very important to have this cold active enzyme, because they can actually use it without uh, uh, need more energy. Yeah, thermolabile, okay, precisely and easily control. Yeah, minimize the loss of volatile compounds, minimize the indesirable chemical reactions. Yeah, and as I mentioned, okay, it's at low temperature, so we don't need more energy. Yeah, and so it can work at a lower activation energy, so that it cause the productions. Yeah, and in terms of the specific and selectivity activity, it shorten the processing time and reduce the concentrations of the enzymes. Okay, so this is the potential of utilizations. Okay, we can use it as a in a bioconversion process. Okay, detergents. My project currently is dealing with detergents, yeah? Using the cold active enzyme. Okay, so we link it with using the clear cross linking. Yeah, so we want to use this detergent, enzyme based detergents, that can be used in the cold temperature. Okay, so you don't have to increase the temperature, because if you go to cold country, yeah, so that when you want to wash your clothes whatsoever, you need to have a what I call the cold, sorry, the hot water, not the, so they need to increase the temperature, so that it can the enzyme can work. But with this enzyme, okay, we can just use at a low temperature. Okay, in terms of fermentation, paper and pulp, okay, for molecular, of course, molecular work, detergents, the cheese productions, and textile, food and textiles. Yeah. All right. So this is novel enzyme, which has been isolated from Antarctica. There are a few only. Yeah. For examples here, lipase and esterase. Okay, they screen using the agave assay. Okay, in 2011 at Terre Adélie. Yeah, the Pointe Geology Archipelago. Esterase at McMurdo. Yeah, this is American base. Yeah, from soil using the agar base. Okay, and lipase at the King George Island. Okay, also using the agar base assay. Chitinase again at the King George Island. but this one is a lake sediment using the gene sequencing, and alkane monooxygenase, okay, at King George Island. Because if you want to know that King George Island, there's a lot of stations, Antarctic stations over there. That's why many many studies have been done in King George Island, right? All right, so this is my approach. Yeah, I use both culture dependent and culture independent, meaning that I do the plating and also the metagenomics analysis. Right. All right, so this is the sampling site. Okay, near the McMurdo U.S. stations. Okay, what we call the Ross Island. Okay, this is this has been taken. The sample is around 2008, 2009, something like that. Okay, we collected the samples there. Okay, I've been there to Antarctica, but not this area. Uh, but at the West Antarctica, the remote islands. Okay, we call it Greenwich Island at South Shetland archipelago. Okay, as you know that Antarctica is a continental, okay, continents. So it's not like Arctic. Arctic is a frozen sea, yeah, but Antarctica is one of the continent. So they have these volcanoes, rivers, and all that, but covered with ice. Okay, so there's a lot of opportunity to be there. Yeah, if you join, if Unisel perhaps in the future join the Yayasan Sultan Mizan Antarctica Research, okay, because UIA is not yet joined the partnership. Okay, I get this ticket before they introduce the partnership program. Yeah? All right, so maybe some of you can also join the Antarctic project. All right, so these are my samples. Okay, at near the McMurdo. Okay, specifically at southern Victoria Land. Okay? We have we need to check the descriptions of this sampling site. Yeah, because later these things will actually influence the type of the bacteria that we isolate. Okay, or we get it. Okay, I have 7 specific locations. Okay, Botany Bay, Cape Royds, Dunlop Island, Marble Point, Cape Roberts, okay, Cape Rose, and also Spike Cape. Okay, this is the place. If you want to see, this is the small box there. That is the locations of the sampling site. All right. So the first approach, yeah, culture dependent. Okay, we screen using the skim milk because we are looking for the protease. Yeah? So it's a normal what I call the plating techniques, but you make the plate using agar and skim milk. Okay, the formulation mix like that. Okay, once you have this, once you have the bacteria that produce the protease, it will form the halozone. Yeah? So this is the positive indicator, okay, that the bacteria colony can produce the protease. Okay, and then we test it, okay, using the azocasein, okay, for enzymatic assay. Then once we confirm, we do the sequencing, okay, 16 sRNA sequencing to identify the bacteria. Okay, this is culture dependent. Okay, but I also do the metagenomics approaches. Okay, amplicons, 16S amplicons, metagenomics, and we continue to the functional metagenomics. Okay, so this one we just extract the environmental DNAs. Yeah, but we cannot use for these methods. We tried several methods of extractions using the kits and all that. But it's very difficult to get the DNA, okay, from this environment. So we use the conventional methods of phenochloroform extractions. Yeah, and we optimize the conditions, and then we manage to get good quality of the DNA. All right. All right. So this is the what we found out. Okay. For the culture dependent, okay, we managed to get 46 bacteria. Okay, it's very difficult. Okay, and it take a longer time because we need to incubate it at low temperature. Okay, we incubate at 4 degrees, 15 degrees. Yeah. So it take longer time to grow. Yeah. But we managed to get about 42 pseudomonads. Okay. 3 artobacter and 1 pennibacillus. Okay. Where out of this 46, we found out that 8 bacteria form halozone. Okay, on the skim milk after 24 hours incubation at 4 degree. Okay, meaning that they can actually work in low temperature. All right. So from that eight, okay, which show the protease activity, yeah, we run the enzymatic assay and we found out that SC8, okay, skip spike cape samples is the highest in terms of the enzyme activity, the proteolytic activities. Yeah. All right. From that, okay, we do the once we know they have this proteolytic activity, we do the optimizations of the growth of the bacteria. Okay, we test on we test in terms of the temperature, the pH and all that. Yeah, the optimum temperature for growth rates for all bacteria is 20 degrees Celsius. That's why I mentioned just now. Okay, this optimum, so meaning that they are cyclo tolerant. Okay, they can go further. Okay, at more than one of the samples or the isolate can go up to 40 degree. Okay, they can grow at 40 degrees, so they are cyclo tolerant. Okay, so this cyclotolerant, okay, as I mentioned, it can go up to 4 to 36 degree because they are facultative cyclophiles, but their optimal growth at 20 degree Celsius, right? Okay, so the next optimizations, okay, we use OFAT, one factor at time. Okay, the second factor is the pH. Okay, so uh, we found out that this isolates actually in terms of where we get the samples, okay, will affect the pH and also the temperature. Okay, for examples here, the isolate BB and ROB, okay, we collected at the Moses community. Yeah? Ross at algae and cyanobacterial mats. Okay, so they have a lot of organic matters. Okay, and SC is a abandoned squaw nest. Squaw is a kind of bird. There are one or two or three birds only. Okay, type of birds in Antarctica. So squaw is the dominant one, I think, for the Antarctic. Right, and salt. Okay, sodium chloride, because we collect our samples near the shores. Okay, so that the salinity will actually influence the distributions of the bacteria. Okay, and all bacteria strains showed maximum growth at 4% sodium chloride. Okay, this is according to the seawater concentration is about 3.5. Yeah, it's near there. Yeah, and when we collect at shore, because of the wind effect and all that, it will bring the salts, okay, to the terrestrial area. Yeah. All right, and this is activity of proteolytic assays. Okay, we found out that this SC8, okay, is the best. Yeah, and also Ross. Yeah. When we compare with the blue color, the protease, the standards, okay, we found out that at the low temperature, okay, SE8 and Ross, okay, will give the higher activity compared to standard protease, okay, up to 20 degrees Celsius. But after that, okay, we can see that the protease, the standard protease, will show the more activity compared to the cold active enzymes. Right. So once we have this, okay, we can see that as SC8 is the highest at the low temperature. So we select SE to further what they call further optimize. Okay, we use this the growth rate. Okay, so we design, okay, using RSM and all that. Yeah, so this is the design expert, right? So we have 20 treatment for this. Yeah, and we found out that the bacteria growth rate is varied from 0.0195 to 0.671, yeah, per hour, right? And this is the optimizations via RSM, okay, and this is only for AC8. Yeah? So when we run the ANOVA, okay, we found out that okay, the lack of it is not significant, meaning that this model is good, right? And R squared is 0.95. It's also good. Yeah? And the reasonable between the predicted and treatment, okay, is 0.2, so acceptable. Yeah, in terms of the statistical analysis, whether the model or the design that we use is appropriate or not. All right. And from the model, okay, it suggested that okay, the 2.05% of sodium chloride concentration, okay, can be used for the uh, better growth of SC8, pH 6.83, and temperature at 20.5 yeah, degrees Celsius, right? All right. In terms of the non-culturable, okay, amplicon metagenomics. All right. So we proceed with Rob, Ross, and AC. Okay, and we found out that this is uh, the what they call the phyla. Okay, that we found. Yeah. You can see that most of it is the planktomycetes and also the proteobacteria. Yeah, most of them in all three sites. All right, and we can find that this Rob, okay, unique OTUs at Rob is 476, Ross 552, and also the SC is 601. And between these three, only 15 OTU shared by these three sampling site. Okay, meaning that they have a similar what they call phyla for each of these site. Yeah. All right. And this is the bacteria that the strain that we find, okay, from these three what they call three sites, okay, all refer to uncultured bacterium, yeah, and only one that we can actually confirm the strain Spingo hebus, yeah, right. The dominant OTUs, okay, we found out that this dominant OTUs is at ROB, yeah. From there, okay, we run the functional metagenomics, yeah, so that we can find what is the potential genes that express, yeah. And you find this is the functional metagenomics data. Okay, in the middle is actually from the host cell, yeah, and what we want to find is at the outer layer, okay, without outside of the round area. Okay, we found out that node 50, 64, 67, and 69 is still part of the E. coli mobile genetic elements. So from that, we only left node 42. Okay, so we take this node 42 and we analyze further. All right. All right. This analysis of node 42 is about 35 kilobase pair. Consists of 56 contigs, 53 hypothetical proteins, and they have the specific target contigs about 893 base pair. Okay, and when we run the PFAM database analysis, we found out this node 42 consists of peptidase. Yeah, and we run the Swiss model. Okay, a bit bioinformatic analysis. and we found out that they have this histidine motif, okay, which indicate that this node 42 consists of this peptidase. Yeah? All right, so we ran the homology modeling. Yeah, it found out that we have this 23.29% similar to the 3-SLU, yeah? with RMSD about 0.402 Armstrong. Yeah, in terms of the amino acid compositions, it consists of lower arginine residues and higher proline compared to 3-SLU. Okay, and we found out that they have this large deletion sections. Yeah, and the structure is more flexible, but it induces fit theory. Okay, it's actually before we run this experiment. Okay, we also have preliminary data. We found out that this is true. Okay, where we combine all the structure in the database, okay, from the temperate and cold active, and we do the comparative analysis in terms of the structure. We found out that the active sites actually there's a lot of deletions, yeah, in terms of the active site. Okay, so this is supporting, okay, supporting the findings, the prelim studies that we do in terms of the structure. All right, so for the conclusions, okay, we managed to get, I can say it's a novel, okay, novel enzyme because it's only 26%, okay, similarity when we compare what in database, okay, but we need to go analyze further. We are now working with UPM, okay, to actually do the X-ray crystallography, okay, and after that we will do the what I call the upscaling in terms of the enzyme production, yeah, and then we need to further purify. All right, so as I mentioned, in future the purification and characterization of this enzyme is essential, okay, in order to identify the optimum working condition. Okay, at this current moment, my student is doing the cross-linking to make use in terms of the detergent products. Okay, so with that, thank you. Assalamualaikum warahmatullahi wabarakatuh.
Thank you, Dr. Faizul. That was an interesting and inspiring share. We open the session to Q&A.
Any questions?
Okay, Assalamualaikum, Doctor. One question.
Yes.
Doctor, among all of the tasks and all of the lab work, okay, that you have been conducted there at the coal region, what are the main challenges, Doctor?
The main challenges.
Thank you.
Okay. First is the facilities. Okay, it depends on where you was stationed. Okay, some of the station, the lab equipment is very good. Okay, but in terms of my station, I was at Arturo Prat, okay, a Navy station where we only have one small lab with basic instruments, so we cannot do much. So my work is just I collect the samples, I extract, and then sleep. Right? So it depends. It depends. Yeah, it depends on the facilities. If you go to the high-end stations like King Sejong, Korea, or you go to US stations, yeah, British station, they have a lot of facilities. Okay, but I went with Chilean. Okay, so the facilities is limited. Currently, we don't have our own station. Okay, and I think last year they opened applications for winter expeditions. Because if we want to have a station, okay, we need someone that can take care of the station during winter. Okay, this is the first time last year I think from UMS, Prof. I'm not so sure. Okay, because you need to be there more than 6 months. Okay, you enter before winter, and then you go out after winter. So it's like 6 months, something like that, to be in the station. So it's very hard. So we are trying to get the station there. This, eh? My work's about 2 months. 2 months during summer. Okay, during summer is about 2 degree to sometimes can go to -5, -6. Winter is about -60. Okay. So for students, okay, if you want to go to Antarctic, uh, challenge yourself. Okay, then you can involve with this project.
The audience could still shoot one or two questions to Dr. actually.
Yes. Okay, all right. So when we collect the samples, that is easy part. Okay, the difficult part is to bring your sample back to Malaysia because we need to get the permit. Okay, from Department of Agriculture because this is soil samples. So it take me about 1 year. Okay, because I'm the first one in the Yapazam or Yayasan Antartika that bring back the samples on my own. Okay, before this they bring the samples using the agent, okay, the company. So everything they settle, the company will settle the permit and all that. So it's quite difficult, okay, because we need to get the permission. Before get the permission, we need to. They will inspect our lab. Okay, they don't want any vectors to enter the lab and go out, so that maybe it will carry the dangerous germs or whatsoever. Okay, so once we have that, we need to specify the specific freezer. Okay, how to where to keep the samples. Okay, in terms of the working, because mine is more on cyclotolerance. Okay, we can work like normal. Okay, in the laboratory, but my lab is the aircon should be the lowest, 16 or 18 degrees. Okay, if I and some of because I have two rooms for myself, so one of the room I use for my office. One of my office I use for keeping keeping the culture there. Okay, is everything is in a cold temperature, not more than 20 degree. Okay.
Yes, Dr. Fazal, I have one question here.
Yeah.
Thank you very much for your sharing session. So your talk just now remind me of my master project, which I'm also involved in metagenomic studies, which working with the metagenomic sample from seawater from Pulau Tioman. And I understand it's difficult to collect the sample because the station is very limited. So collect the sample. The most important is how to preserve the sample until getting back to the lab. Because unless you don't have, because you have a very limited cold storage, so how you preserve the sample until you get back to the lab. So that's for my first question. Because otherwise when you come to the lab there's nothing inside there. That very and make things even worse, that you might like lost the potential of all the microbes that you look into it. So that's my first question, how you preserve the sample. The second thing is what's the because working with the what do you call this antithetical samples and then come back to the lab, I mean with the temperature of let's say 30 something. So what was the challenge like getting this like this cold protein, because it's not easy, right, to um, express this cold protein in normal temperature. So maybe you can share your experience on that. How you overcome the obstacle. And lastly, going to the future, because currently we have like the nanopore platform, which helps you to analyze the metagenome sample. During our time, I mean, like during 2000, before 2004, we working with using a culture method. Now, I don't think people are doing that anymore. So they straight away jump to the using a NGS platform to sequence. And now we're having a nanopore platform. So I believe it will help to make this metagenomic studies cheaper and also become faster. So maybe you can share your thought on that using this third-gen platform on the metagenomic studies. Thank you.
Okay. All right. Thank you, Doctor. All right. So in terms of the bringing back the samples, how we preserve in terms of the quality of the samples. Okay. When we bring from Antarctica to Chile, okay, we use the Navy ship. They have the -80 there. Okay. So we just put inside there. All right. Once we arrive at Punta Arena, okay, Chile, and because I have a collaboration with University Magallanes, okay, just at the near the port, so we straight away bring to the University of Magallanes. Okay, from there to Malaysia. Okay, that is the issue. One is the samples permit. Okay, because I bring it on my own, so we need to have this, the cold pack full of the cold pack and all that with all the soil samples inside there. Okay, every time I stop at the airport, I have difficulty in terms of the security check. Even to bring to, you don't carry, right? You just go to the baggage. So every time I've been called for the security check, because when they go through the scanners, okay, it will show you all blue because it's cold. And they ask to open it. We show them our permit, okay, both from Malaysia and also permission from Chile. So if you want to go next time, try to find the less what they call transit. So because I choose the cheaper one, okay, like macam kapal terbang lompat-lompat. So yes, because the budget is small, right? So we need to face that, the challenge. Okay, once we reach the what they call the airport, okay, we need to store that in markis. Okay, markis, they have their own -20 before they take the samples and bring it to run the test first. Okay, the test in terms of the bacteria, virus, what they call the larvae whatsoever. Okay, all types of tests to avoid the pathogenic, okay, or the pests. Okay, from the samples. Okay, once they clear, then I bring back to my lab, all using the cold box. Okay, right. So the question number one. Question number two, in terms of, yes, last time we use the NGS big machines whatsoever. Now we have the nanopore. Yep, it's good to have that. Very small. Use the laptop. You can bring straight to Antarctica. That is preferable. In terms of what they call the 16S amplicon metagenomics analysis. Yes. All right. Okay. Okay. Any question?
Thank you, Dr. Faizul. I would like to invite Dr. Muqtazul Haizat, Deputy Dean of Academic, accompanied by the Program Director, Dr. Yashoda Sundaraj, to present tokens of appreciation. Please join me in welcoming them to the stage.
We invite our keynote speaker, Associate Professor T.S. Dr. Nur Faisal Hadrinodin, to accept the carefully prepared package of gratitude. We request that Dr. Faizul, Dr. Haizad, and Dr. Esoda to remain standing. Before we proceed to the poster presentation, we would like to capture this moment with a group photo. We invite all VIPs and panels to front the center and for all the attendees to gather in the middle for a quick picture together. Your participation will help us mark the beginning of the exciting event. Thank you, and we appreciate your cooperation. Please be stand at the front, the center, facing the screen, and everyone, please gather at the center.